After getting his Ph.D. at the University of Newcastle, went on to the University of Guelph, where he spent the last 10 years or so working on optimizing greenhouse floriculture and vegetable growth. And recently I noted that Dr. Zheng won the Research Award of Excellence for the Green Roof Initiative at the University of Guelph. And his work is really centered around optimizing nutrition and efficiency in controlled environment growth. So, Dr. Zheng?
Thank you very much, Melanie, for your introduction. Hello, everybody. At the University of Guelph, one of our research focuses is plant production under controlled environments, including greenhouses and other indoor environments. We have been working on lighting in horticulture application for years. Maybe some of you have read our papers on moving lighting and comparing different conventional lighting technologies in the past, and then greenhouse lighting environment under hanging baskets, all of this research. But today we are going to be focusing on LED application in cut flower production. As you know, that LED has a high energy efficiency based on a peer-reviewed paper by Bogate, 2008. LEDs were more efficient than incandescent and fluorescent lighting, and on par with HID lamps such as high pressure sodium lamps as early as 2008. LED technologies are now in a period of rapid development with dramatic increases in energy efficiency and then decreases in price. Before 2008, their efficiency was increasing 30 times per decade, with a corresponding 10 times decrease in production cost. This trend continues, as stated in a paper by Pinhole et al. 2012, just recently, that the electrical efficiency of red LEDs will double that of HPS lamps by 2020. Number two, as you know, that LEDs have much longer lifespan. For example, most of the LEDs can have a few times longer lifespan than high pressure sodium light, which are currently used for greenhouse production. And number three is their capability to provide specific wavelengths, the light spectra. This is extremely useful and exciting. We know by changing light spectra and the ratio of different light spectra, we are able to modify plant morphology, for example, to get plants grow taller or to change the elongation, the plant growth speed, even change phytochemical contents, etc. For example, recent research showed that right LEDs can be used to produce compact poinsettias and then control the height of certain bedding plants very well. Since the middle of the 1990s, especially in recent years, many studies have been carried out in different parts of the world investigating the applications of LEDs in horticulture and showed some very promising results. However, there is still tremendous potential, need further research and exploration. In theory, we know they're going to work in many ways, but really lots of work we still need to get it done, to get it approved, or to develop even protocols how to use these LEDs. Number four is LEDs are dimmable. and then they can be rapidly cycled between on and off, which means that they can flexibly supply light according to outside natural lighting level and then the plant's needs. This way we can further save energy consumption. Of course, there are certainly more advantages, but I'd better stop here and start to report to you about our recent studies. As you know, that cut gerbera is one of the few only cut flowers we are able to produce in North America to make a profit. Wintertime, growers in Canada and most of U.S. Parts do need to provide supplemental lighting. Otherwise, both yield and quality of these cut flowers are not going to be acceptable. Majority of the supplemental lighting at this moment are still high pressure sodium lights, HPS. The question is, can we use LEDs to replace HPS? So to answer this question, we have conducted trials in our greenhouse. So basically, we used HPS lighting 400 watts, and then LumiGrow's Pro 325 blue and red 4:1 ratio. So the trial was set in our greenhouse. You will see here, the greenhouses we have probably, it's not that many people have this facility. As you know, that lighting research is very difficult, especially in greenhouses, because for biological research, we always have to have enough replicates. But how do you block this light spectrum from another one? How do you isolate them from the natural light? Sun is everywhere, you know. So at the University of Guelph, we have a greenhouse lighting research facilities. So you see here, we have benches, all of these benches. Each bench have these curtains. These curtains can automatically coming down right to here or in between. So in that case, each bench, we can have one light treatment. And in each bench, we can treat it as one replicate. And we have many of these benches. So for this trial, we set both LED lighting and HPS lighting PAR level at 60 micromole per square meter per second. This light level was determined by, we went to many, or not many, just a few greenhouse growers to measure their light level in the night when there is no natural light, to see what's the supplemental lighting they are using at this moment. As our other research project, we want to make our research results useful. That's why we went to the local growers, measured, and then came back and set it all the light level 60 millimole or micromole at the canopy level. And then we have 4 different lights, and each one have a few replicates, which means a few benches. So here you will see the daily light integral under HPS lighting. This is natural lighting. This is supplied by HPS. This is LED lighting. Of course, this is from natural light. That much is from the LED lighting. So basically, they are very similar. And then, so here are the results. During the production, the trial period, we harvested every single cut gerbera flowers, and we measured their quality, including mean stem length, their flower diameter, and the fresh mass of each flower. And we used 3 cultivars. You can see one here, one here, one here. And then So here, for example, if we look at stem length, and almost there is no difference between HPS and LED for these two cultivars. But for Terra Sapphire, the flowers under LED produced significantly longer stem length. And there was no difference on the mean flower diameter, but again, the mean fresh mass of the flowers under LED were higher for this cultivar and for this cultivar. So simply put, the quality was better under LED lighting. And again, growers, when they produce flowers, yield is very important. So if we look at this cultivar heat wave, here is accumulated marketable flower. So we harvest each week, and then they accumulated. You can see here the treatment under LED lighting were significantly higher. So more cut flowers were harvested under LED treatment than under HPS. And here is another cultivar which didn't show that much difference, slightly higher, maybe not significant. But for Terra Sapphire, you can see again the accumulated flower numbers produced over these 16 weeks were much higher than under HPS. Also, we did some vase life trial. That means after we done the research, we put them into vase just to see these flowers produced under LED versus HPS would be any difference. And seems to us is that this cultivar, the flowers last much longer if it's produced under LED lighting. But for these two cultivars, they were not that much different. And this is just water usage of these flowers in this vase. So I guess you don't need me to conclude from this trial that it's very obviously that LED is able to replace HPS and then produce either better or the same quality or yield of cut gerberas in greenhouse production. And currently we are doing another trial. We've been talking about LEDs, they are more efficient. And so down the road, we got growers asking us, okay, so if the LED lighting, they are more efficient, energy more efficiency is getting higher and higher. So if we want to provide supplemental lighting, use LED, how high we can go? Because the energy consumption gonna decrease. So what is the best light level we can go so we can achieve the best quality and yield and then also profitable? So in the same greenhouse lighting research facility, we are doing this research, just trying to figure out what is the optimum lighting level for snapdragon, cut snapdragon. So you can see here my research associates, David Llewellyn and Yun. And so this trial going to be finished very soon, and we're going to report our results to you very in the near future. And I guess I'm going to stop here, but before that, I would like to thank International Cutflowers Growers Association and then Joseph Hill Memorial Foundation and LumiGrow for their support, financial support, and providing lights, lots of support. Without their support, we couldn't get this done for sure. And then also, we like to thank our local growers. We always pick their brain. They give us plants. For example, Rose Flora is very, very supportive. And then finally, but not least, the list is my research associates, David, Catherine, and Yun. And thank you very much for your attention. And here is my contact number information. If you have further questions, either you can contact me or George or Melanie. Well, thank you so much. I will stop here, switch to Melanie and George.
Okay, thank you so much, Youbin. Okay, let's see. I'm going to talk just a few minutes about my own work, and let me just get this up on the screen here. So I'm going to talk a little bit about the in-house experiments that we've been doing here at LumiGrow, and also I'm going to try to highlight some of the things that I just heard last month at the LED Symposium at the University of Arizona, and then just finally conclude with some of the applications that we're optimistic we can do with spectral science. So my next slide, so last mid-February, there was a symposium at the University of Arizona, which really highlighted the culmination of a USDA grant for the past 5 years. And scientists came together and presented their work where, how LEDs can be used. And again, I'm just going to highlight a couple. But
you want to go back?
No, it's okay. So this is just some experiments that I did with zinnia in the greenhouse. I used white zinnia and grew them under different light conditions, different ratios of red and blue light. And I'm just showing you a portion of the experiment. You can see on the left there was no supplemental lighting. In the mid was a mid-blue range, supplementing with about 16% blue. And on the far right is our traditional high-pressure sodium. And what I want you to see here is that we could create a very compact, tight plant with a little bit darker green foliage in the mid-blue range. And as a complement to this experiment, I did experiments in the growth chamber. The results were similar but slightly different, again, growing at different ratios of red and blue light. And I was able to create a plant that bloomed when we wanted to. One interesting factor was the variation that I saw in the root production. With a higher blue, we typically got much higher root production, which might be important to people who are doing cuttings or propagation. On the right-hand side, you see a photograph of some plants that I just let go for an additional month after I typically ended the experiment, just to see sort of at a gross level what happens when you leave your plants under fluorescence for a long time versus under the LEDs.
I know which one I would send to my mother for Mother's Day.
Yeah, right. And these are some slides that I borrowed from Dr. Eric Runkle. He's a professor at Michigan State University, and I urge you to go to his website where he has a lot of experiments that he's done looking at lighting over the last 15 years in greenhouse work. And these experiments looked at varying photo period using LED lighting. And what I want you to see is what's really obvious is the variation in flowering, the size of the plant, again, the control that can be suggested with light alone. And like Youbin said, there has been some recent poinsettia plants. This really speaks to whether or not we might be able to start to move away from using plant growth hormones and simply use light to control plants commercially. Okay, I'm going to move now to talking a little bit more about vegetables. In this slide by Cherry Kubota and Ricardo Hernandez, you see a similar sort of phenotype that you get with vegetable plants. They did experiments with tomatoes, cucumbers, and peppers. Also in the greenhouse on the left, in the growth chamber on the right, they were able to control the hypocotyl length and get the phenotype of the kind of plant that they wanted, whether they were doing a cutting or looking at creating plants for the field, etc. Okay, in the next slide, this is where I want to, again, continue talking about our vegetables and plants that we eat, but I want to talk about the health-increasing phytonutrients that we're seeing in the last couple of years. This is a paper by Dean Kopsel and Carl Sams out of the University of Tennessee with Robert Morrow of Orbitech. And in these experiments, they were able to treat with varying levels of blue light and get increase in the carotenoids. Similarly, in the next slide, Michael Jakovich and Carrie Mitchell out of Purdue, they did similar experiments, again, looking at broccoli microgreens. They've done this with kale to see an increase in the anthocyanins and the glycosinolates. So on the right-hand side, not only are these pigment proteins being found, but the volatile compounds are also can be controlled by light. And so Kevin Folta's group is working a lot on that and has seen the evidence that you can affect the aroma of petunias, tomatoes, blueberries, and strawberries can be manipulated with light. Okay, in-house, what I've been focusing on is looking at flavor. And so this is a little tear sheet from our website, which you can look at, which looks at how basil can be, we can change the flavor of basil with the lights. And what you can create is very sweet basil or a rather spicy, bitter tasting basil. And in the next slide, this is a similar type of experiment where we did this with a Salinas lettuce and a black-seeded Simpson lettuce. I got the same results with both cultivars. And again, you see in the pictures there, you could get a flat leaf, very sweet lettuce, which was really preferred by half the people. And on the right-hand side, you could get a curly leaf, arugula-tasting, spicy lettuce leaf. And I just real dramatically showing what we can do to steer plants to be what we want them to be. Okay, so far I've talked about young plants and haven't really talked about what's happening in commercial production. Certainly, Carrie Mitchell and Selena Gomez have done some really nice experiments looking at how high-pressure sodium is comparable in the production to LEDs. And I think that, George, do you want to speak on some of our commercial production people that are happy with the lights?
Yeah, sure. So, we have over 3,000 installations in 30 countries from some of the top growers, agribiz, and more than 200 universities. And, we've got trials out in the field with different commercial growers. And this was a trial that was recently completed that showed that for mini cucumbers in a greenhouse with supplemental lighting, our LumiGrow LEDs were able to produce a 67% higher yield using 56% less electricity. So on a kilogram per kilowatt hour basis, we were able to outperform high intensity discharge lights. And this was after, the greenhouse had calibrated the various inputs and variables inside, including temperature, humidity, their nutrient levels, so that they had created the optimal environment for the LED bays. And when we compared the two, we were really happy with the results, and the grower was ecstatic.
Thanks, George. Yeah, and I think that this is coupled with the fact that the cost of LEDs keeps going down further and further. I know our lights are cheaper than they were in past years, and so the previous sort of thought that LEDs are too expensive, I think, is really changing as LED costs drop. So what I want to leave you with today is that what we call spectral science, the science of optimizing plant growth with light, is just an exciting thing that we're just embarking on. And among the elements that we can control are taste, nutrition, making the anti-cancer compounds that we'd like to have in our foods, morphology. We can have a short type plant that's not flowering or a tall skinny plant that is flowering. Fruiting, the time to fruiting, and we can also affect the quality of our fruiting and time to harvest. So I think there's a lot of exciting work to be done. So do we have time for questions?
Yeah, we've got about 5 minutes for questions, and we can stay a bit longer if there are a lot of questions. To submit a question, you can use the question panel and the control panel on the right side of your screen. And if there are questions that we can't answer in the time we have today, we'll get back to you by email. And meanwhile, while the questions are coming in, and as I'm fielding them, I'll be just showing a little slideshow of some of the installations that we have around the world. Here's a question. Someone's asking about the impact on water usage. Now, you've been showed some slides, and Melanie, I think you've seen some anecdotes in the field about the effect of water use under LEDs. What have you noticed in terms of water efficiencies?
Most of the data that I have are from growers using high pressure sodium, and water usage depends on a lot of things, and a plant of a certain size is going to use the same amount of water of the same plant of a certain size. But what we see most is because of the radiant heat of high pressure sodium that LED grown plants tend to use about 20% less water in greenhouse grown plants.
Excellent. I have another question here from a grower, and the grower is asking how they can calibrate and use spectral science to optimize their crop for their particular market. Do you have I suppose this is sort of similar to structuring an experiment. Like Melanie, what would you advise to the grower?
Yes, there is a transition time where you have to figure out, what's right for your plant. I wish there was a magic bullet where I could say, all plants behave in a certain way. We do have information depending on what you're growing that, I can certainly provide people with if you're growing, peppers or tomatoes and what's worked with a certain cultivar. But I would recommend that, you be mindful that there is going to be a transition in learning how to use LEDs and change your water regime, things like that.
Professor Youbin, do you have anything to add?
Well, I agree with Melanie. It depends on your crop. For example, if you are growing bedding plants, you don't want the plants to be too much stretched when the greenhouse lighting level is not high. And then you can change the red and far red light ratio to control the height of your crop. But then if you are growing nutraceutical plants, and then there are certain spectrum, even you can use UV to change or enhance some of the phytochemical content. So yeah, exactly as Melanie said, it's case by case. But the good thing is that now we have the tool. LED is able to provide all of these requirements, because in the past we just couldn't. People, you remember, people were using filters to filter out these spectrum and that spectrum, trying to manipulate in research the plant's response. And now that's why I said it's exciting. Yeah.
Yeah, it is definitely an exciting time for research in photobiology. Along those lines, we have a question on cucumbers. The question is, what is the best blue-red ratio for cucumbers? Melanie?
I would urge you to pull up Cherry Kubota's talk from the University of Arizona. And I'm not sure if I'm assuming this is a greenhouse grown circumstance. And in her work, she found that actually using 100% red as a supplemental light was recommended for cucumbers. I will also comment that in her work that she used 2 different levels of DLI in the greenhouse. And I think it brings up what Youbin had brought up about how the lights can be dimmable. So as we get better at being able to assess the DLI coming into the greenhouse and knowing exactly how much red light or blue light to complement at that DLI, because what she found was that high DLI versus low DLI, you got different behavior and a different need for supplemental lighting.
Another question we have here, are there any light recipes for improving rooting in seedlings? Again,
in my hands, I got an increase in rooting pretty dramatically with 60 micromoles of blue light with the zinnia. Ricardo Hernandez, he has a nice talk at the Arizona Symposium on rooting. and getting increased root production.
So it does sound like there are light recipes that improve rooting for seedlings, and it probably is dependent on crop and cultivar, but I think that information is out there, and certainly there's more information out there to be discovered.
Yeah, one thing, George, that we're just putting together here at LumiGrow in research is we're distilling a lot of the papers and information that's out in the literature and trying to create an open source database where people will be able to come in and enter their crop that they're interested in and really get a distillation of all the information that's available.
Yeah, we're excited. We're going to try to build a database of LED research that's keyword searchable as well as tagged. So we're hoping that will help scientists and commercial growers kind of zone in on information. Here's a question about the change from HPS to LED and the adoption in commercial environments. How is the transition taking place? And is there a huge acceptance of the technology, of LED technology? From what we've seen, when we talk to growers and we're able to speak with them about the effectiveness of our fixtures and show them our research briefs and case studies and give them examples of our growers, we get a lot of excited growers that engage with us. I think from a wider scale, there are a lot of myths out there from the LED industry, and Professor Zheng, you may have seen it too. There's some, in the early days of LEDs, before they were powerful enough, they were also too expensive. Some of the trials didn't go so well, based on the marketing hype. But I believe that the industry is now poised and is poised to make the transition over to LED lighting because high precious sodiums, let's face it, and I think everyone would agree, they're energy intensive, they're wasteful, they do provide a lot of light, but you also you don't get the level of control. So it'll take a little bit of time to transition the industry over. It'll take a lot of education to do that because let's face it, a lot of growers and people are creatures of habit and they stick with what they know. But I believe moving forward, given the state of the world and the industry, we should try our best to accelerate the adoption of LED technology in greenhouses and growth chambers.
George, I noticed that question came in from Mexico, and I just want to point out that as the energy cost does drop and growers are seeing the benefit of lighting, that we're seeing the use of lighting further and further south. And we just had a really successful tomato grower in North Carolina who'd never used lights before and just so excited about their winter production. And it's a situation where if you haven't gotten attached to high pressure sodium, I think every grower I've ever worked with that starts with LED and they're not transitioning from an old way to a new way, but are simply implementing LED for the first time, they're really pleased with the results. So LED lighting is allowing lighting further south and more production.
Here's a question. Is this research only applicable on greenhouse plants? In other words, is the research only applicable for herbaceous plants, or can it be applied with potted fruit trees growing under greenhouses as well?
We have actually several growers who are doing potted trees. Let me think. The one that comes to mind most is citrus growers who start their trees in the greenhouse and then hard them off in a lath house or something before they go out into the fields. And we have several customers doing trees.
Let's see here. We got a couple more questions. Time for one or two more. Do we have any guidance on how to use LEDs to improve post-harvest shelf life? Professor Zheng, you found some results with regards to the marketability of flowers. Maybe you can answer this one as it relates to the research you've done.
Well, as I showed you, the results that some of the cultivars, gerbera cultivars, performed better for the waste life. But then if you want to provide guideline there are quite a bit of research to be done. So I don't think we have enough knowledge to provide a guideline at this moment. But the results we collected just looks very promising. And we even don't know what's the mechanism of why it performs better.
Yeah. Here's another question. Where is the technology gaining more acceptance, in vegetables or flower crops?
I think it's both.
Yeah, I would agree.
I think it is both, really. Vegetable part, you look at basil and some other nutritional, the vegetable have nutritional value. You are able to either produce it faster, or you can change the color, and you can improve the nutrient content. And then for flower part, as well as Melanie mentioned that, you can control height. Height control is a huge thing for ornamental production. So really, I think it's got potential in both.
Yeah, I would agree. I think in terms of the market and what I've seen on the marketing side, it is both flower production, vegetable production, medicine, nutraceutical production. Vegetable growers tend to be quicker to buy for some reason. But flower growers, we have a lot of growers. In this case, this is a large flower grower in Half Moon Bay that's using our lights and trialing. So yeah, we're seeing a widespread adoption throughout all the industries, and that's really exciting to us. It shows that the economics are there, everything pencils out, and that that folks, based on the trials that we've seen, the successful trials, that the commercial growers are really keen to take on the technology to improve their production. Let's see. I got time for one more question. Let's see here. Just a moment. Okay.
Well,
yeah, go ahead.
Oh, George, I just was going to comment and add to say that, when you're talking about, shelf life, or we don't have as much data as we'd like to have on that. I would really welcome if anybody would like to collaborate on that type of work. But we do see is we have seen several reports of lower powdery mildew or fungal infestations of the greenhouses or growth chambers under the LED lights. And again, there's no definitive work that I've seen. I've seen one paper on that, but there's a suggestion that LEDs have some inhibitory effect of fungal infection and maybe some other pest.
Cool. All right, here's our last question, ladies and gentlemen. This is from Jamel. The question is, are there any results on fruit quality parameters using LEDs that can be shared? Well, I know that we've got some trials underway looking at tomato flavor. We're basically analyzing pH, the acidity, and the Brix count and sugar content. There are other research papers out there that look at the taste and flavor and fruit quality as well. So yeah, the literature's out there, and maybe what we can do is send you some links and share those with you. At this point, I'd like to thank Professor Youbin Zheng of the University of Guelph for offering to share his research and spend some time with us. And I'd like to thank Dr. Melanie Yelton for pushing forward the idea of spectral science. It's as we're very excited about the direction of the industry now. The technology is here to support the use of light, and the economics pencil out, and we couldn't be more excited about the progress of the industry.
Yeah, thank you, George. Thank you, Youbin. And I urge you, if you have any more questions, we're going to be having a Reddit next week, an AMA, Ask Me Anything, on Thursday, March 12th. The case studies that I've mentioned, as well as a summary of Dr. Zheng's research on gerbera, is on our website. Let's see, it looks like you can follow us on social media. And also, I am looking for people who'd like, if they're interested in some of the experiments that I've talked about, I would welcome collaborations with either growers or scientists out there who'd like to work together. Thank you.
Yeah, thank you all very much.
Thank you.
Professor
Thank you
Youbin,
very much.
thank you.
Take care. Bye-bye.
Bye now.
All right, everybody. If you do have questions, feel free to contact us offline. We'd be happy to answer your questions. There were some questions that we couldn't get to, so we'll respond to those and send you an email. And we look forward to having you attend our next webinar. So everyone, have a good morning, good evening, and good afternoon. Bye-bye.
Thanks. Bye now.
Bye-bye.